and people who have joined online. So today's, the very topic of this conference, International Conference on Sustainable Utilization of Bioresources, is very much relevant and justified in today's context, because as we all know, this has been a serious issue. I will be highlighting some of the things in a couple of slides in a few moments. Why this sustainable utilization of bioresources has become very important in our day-to-day life. And since there has been a lot of debate going on how to sustain our very own lives in this planet is now under question because of so much of these anthropogenic factors which has been contributing to global warming, climate change, and so on. And one of the important ways how we can address this, it is not about saving the earth. It is about saving our own civilization or the humankind itself, because earth know how to mend itself. Whether that means wiping out the human race from the earth, it will happen. But it is for our own survival. We want to do that. We want to make our environment more green, and maybe reduce our emissions and reduce the pollution what we are contributing to the environment, so that this earth becomes a wonderful place to live in. We don't have another option as of now. We have only one planet. So with that, I will start my talk. This is primarily, I'm talking about the biomass hydrolyzing enzymes for tomorrow's biorefineries. And why biorefineries? Of course, earlier, both our Honorable Vice Chancellor and Pro Vice Chancellor had highlighted about the biorefineries, how these bioresources can be sustainably utilized for generating energy requirements, as well as for contributing to our requirements of chemicals and materials, which currently comes from petroleum resources. If we can replace at least part of them, that much we are contributing to betterment of our environment. So the context of having a biofuel or a biorefinery is not just replacing petroleum for the sake of replacing petroleum. And we all know that, we have been fed this information that petroleum is not going to be there forever. It is a non-renewable resource, and soon, today or tomorrow, petroleum is going to be exhausted. So one of the prime reasons for people to start working on bioresources was this concept that petroleum is going to be over soon, and we need to find an alternative source of energy. But that is not just the case now, and even more pressing and important thing right now is how do we address this climate change, because the burning of fossil fuels is the prime contributor to greenhouse gas emissions in the world. And if we can make it, if we want to really make a dent in that, we really need to address how to reduce our consumption of fossil fuels. So this is the context of biorefinery. Tomorrow's economy would be bio-based, because that is the most important renewable resource available on this planet. And we want to make everything possible from biological resources, which is renewable and green, so that we can contribute to making this planet more clean. Okay, this is just to show how do we stand in terms of our consumption of crude in our country. And okay. So I would not. Am I audible? Yeah. Okay. So what we have here is our consumption of crude oil in India. It has been steadily growing, as you can see from this graph. It is still growing up. And from last year, the current fiscal, if you look at what is the consumption of crude, it has gone almost 200% high. And we are now consuming 74 million tons of crude, which is imported in India. And this contributes, we are spending almost 62 billion US dollars on purchasing this crude. This is a pretty high amount. for our country, because this is an amount we can utilize for other developmental activities if we have an alternative to utilize this, I mean, renewable resources instead of petroleum. But and of course, as I mentioned, a more important and pressing need is how to reduce our pollution. This is a picture taken from internet about this morning in Delhi in winter, where you can see the air. This is not just fog; it is basically smog. So we have a lot of pollution in the atmosphere, and visibility is very less. Several times we see that flights are cancelled because of low visibility, and air quality is worst in the world in Delhi. So these are all happening because of the you know very huge increase in the vehicle numbers in the capital region, as well as, of course, some amount is contributed by burning of uh, basically the straw in Punjab, which recently has been addressed. So I will not uh, talk more about that. And of course, our honorable Pro Vice Chancellor mentioned about the microplastic challenge. And plastics we are, discarding into the nature like anything. I mean, this is contributing not just to pollution. This is affecting the lives of every living being around us. So several animals consume this, thinking that this is food, and this is damaging their system. And several times we find that even in forest, deep in forest, we'll find that death of wild animals is happening due to consumption of plastics. So all these has to reduce. I mean, why can't we think about using sustainable resources and plastics, or maybe alternatives to plastics which are sourced from renewable sources, and which is not harmful to the earth, it is biodegradable. So all these are possible if we think about looking at it in terms of converting biomass. Starting from the biomass, if we can make things which are biodegradable, which are green and environmental friendly. And that is where biorefineries becomes important. Analogous to petroleum refineries, you start with a biomass. Instead of crude, you start with biomass. You fractionate the biomass into several components. Most of these plant biomass, what you're talking about, are lignocellulosic in nature. It contains primarily cellulose, hemicellulose, and lignin. And so this is what I was talking about. And this cellulose is a linear polymer of glucose molecules. And you have hemicellulose, which is a branch chain polymer. And primarily, there are two sugars, pentose sugars, which makes up most of the hemicellulose. Of course, there are other components also. These are xylose and arabinose, which are pentose sugars. And you have lignin. And this is a very representative structure. We have fibrils of cellulose, and it is embedded in a matrix of lignin, and interspersed we have fibers of hemicellulose. And lignin is a polymer, is again a branch chain polymer of aromatic compounds, primarily syringel alcohol, sinapyl alcohol, and coniferyl alcohol. So all these things can be converted into very valuable products. Any of these breakdown products of cellulose, primarily it is glucose. Glucose can be fermented to produce alcohol, ethanol, methanol, butanol, or from there you can have glycols and even several different compounds like lactic acid, which can form as a basic starting material for several other compounds. We can have furan derivatives from the pentose sugars like fafural, which can be used for synthesis of 1,3-PDO, 2,3-PDO, and a lot of different, these are platform chemicals which can form the basis of synthesis of several plastics or plastic analogs, which currently comes from petroleum. And lignin, of course, you have a lot of phenolics, and these phenols cresols, and a lot of things which can be used for replacing the equivalents which is coming from petroleum resources. So this is very analogous to our petroleum refineries, and that is why you call it as a biorefinery. So, but for doing this structure, which is a very recalcitrant structure, has to be broken down. This has to be broken down to release the sugars and maybe the monomers of lignin. And to break down this, there are multiple methods. And multiple methods means you can do a high pressure acid digestion, which is the most common and probably the cheapest of the methods. but you require very high temperatures and high concentrations of acid, which is practically kind of very difficult because you need glass line reactors for doing that. And if at all, if it is not glass line reactors, you have to use very the materials which are resistant to corrosion, and you need to use very high pressure to attain that kind of temperatures. So this is all not so easy. And even after doing that, you get all your monomers in a highly acidic solution. And to neutralize that, you probably have to add an alkali like sodium hydroxide. And typically sulfuric acid is used for digestion. And eventually you will end up getting a lot of gypsum. That is basically the sodium sulfate. And this gypsum again becomes another environmental problem. So naturally, what is the other possibility? You can use biological methods for breaking down. NIIST sir already mentioned about using microorganisms, enzymes, and other things. And we are looking at enzymes as one of the most probable or most important means of breaking down these polymers. So how do you do this? When you start with, I'm just giving an example of how do you go from biomass to ethanol. Ethanol, I'm just considering as an example of a biofuel. It can be anything. I mean, it can be ethanol, methanol, butanol, whatever. But typically, we are now talking mostly about bioethanol. So let us look at how this is converted. You start from biomass, and to break down the structure of lignocellulose, we are assuming that we are going to use enzymes. Because these cellulose fibers are embedded in a lignin hemicellulose matrix, it has to be broken down so that the enzymes can access the cellulose. So you have to remove the lignin or remove the hemicellulose. And that process is typically done by using a pre-treatment where you can use a high pressure, high temperature, dilute acid treatment, dilute alkali treatment, or so on. So the lignin is removed, and you have the processed biomass, which is mostly cellulose. And you add the cellulases or hemicellulases, the enzymes which break down this, and you get fermentable sugars, glucose and pentoses. And these sugars can be fermented to ethanol. It can be fermented to other kinds of products. And each stage, there are multiple challenges. As we heard from previously, you probably know that there are challenges in all these steps. And the term sustainability is very loosely used. Sustainable doesn't mean that it is so easy to do, and whatever is planned is sustainable. It is not like that. There are a lot of issues with sustainability. You need to look at the availability, whether the same biomass has other utilization. Maybe if you take out the entire, whatever, suppose you are talking about rice straw or wheat straw. Wheat straw in Punjab is used for feeding cattle, whereas rice straw is not used. But here in Kerala, probably we use rice straw for feeding cattle. And in several places, we also use sugarcane bagasse or sorghum bagasse. All these things, corn stover, all these can be used for cattle feed, or several other applications are there. What happens if you take out the entire thing and use it for biofuels? What will we feed our cattle? So it is not, just by telling that you start from biomass is not enough to have the sustainability. You need to ensure that the biomass is available for whatever current applications are there. At the same time, you can use enough of that biomass to generate viably the biofuels or the chemicals which we plan to get from there. So this includes, the kind of amount of biomass which is available in a particular place. What is the challenges with respect to its transportation and reaching the plant, the process plant, and so on. And pre-treatment has challenges. All these has challenges, but today's discussion, I'm concentrating on the hydrolysis part. And why hydrolysis part? I'll tell you in a short while. So this is based on our own study. We have done pilot scale studies with more than 8 different biomass from different parts of the country. It includes rice straw, wheat straw, sugarcane bagasse, sugarcane trash. We have sorghum stover, we have corn stover, and cotton post-harvest residue. And based on all these studies, we have come to a conclusion. And for all these studies, in the end-to-end operation, starting from biomass procurement and milling to distillation and taking out of the alcohol and dehydrating that, every process was done, and its entire techno-economics have been worked out. So we see that
This is the share of contribution. I'm just giving here the case of rice straw. If you look at the entire cost contribution of different steps to the processing, you can see that hydrolysis step is costing 70%. This is for this particular biomass. I mean, for different biomass, it could be different. But at least in most of the cases, the contribution of enzymes or the hydrolysis step to the entire production of bioethanol from biomass is almost 50%, which means that this is the place which we have to address most so that we make this more viable. So again, I'm just showing some pictures, graphics generated by, I mean, these are, I've downloaded it from the internet because it looks very easy for making people understand how the structure of lignocellulose looks like. So these are the microfibrils of cellulose, and the yellow that you see here are hemicellulose, and these brown ones are the lignin. So now you know that, you know, for an enzyme to access this cellulose, it is really hard to get into this matrix and bind onto these fibers, and it can be more easy if the structure is broken down like this. So this is what when you do by doing a pre-treatment, right? So again, I'm showing how these enzymes are acting. Then mostly the lignocellulose is broken down by cellulases, primarily cellulases. But it doesn't mean that it is only cellulases. There are a group of enzymes, cellulases, hemicellulases, several accessory enzymes, and so on. So I'm talking mostly about cellulose because cellulose is the most recalcitrant material there, which is most difficult to hydrolyze. So this is the typical structure of cellulose material, cellulose fibers. What you see here, all these hexagons, let us assume that these are glucose molecules, which are linked by beta-1,4 linkages. And then there are very organized regions called crystalline regions, and some regions which are loosely packed, which are called amorphous regions. And then cellulase is not a single enzyme, it is a group of enzymes. You have endoglucanases, just like endonucleases and exonucleases, which breaks down DNA. You have endoglucanases and exoglucanases. Endoglucanases creates nicks in between the chain, so that it opens up a reducing end and non-reducing end on the the cellulose fiber, liberating one side reducing end and the other side non-reducing end. Then there are exoglucanases, which acts from either the reducing end or the non-reducing end and starts chopping off pieces of cellobiose, cellotriose, or oligosaccharides. I mean 2-3 units of glucose. And typically it is cellobiose. Mostly it is cellobiose. To some extent there will be cellotriose as well. So one of them acts from the reducing end, which is called the cellobiohydrolase 1. The other one is called cellobiohydrolase 2. These are the two major classes of exoglucanases. And then you also have LPMOs, which are lytic polysaccharide monooxygenases. Very recent compared to the other ones. It's a very recent discovery. This is it is not a hydrolase. It is an oxygenase which acts on the crystalline regions of the cellulose fibers. Again, it creates nicks. But endoglucanases acts on the amorphous region. In either case, it is opening up the channel, the structure of the cellulose, and allowing this cellobiohydrolase to act, liberating cellobiose. And the cellobiose, the final reaction is catalyzed by an enzyme called beta-glucosidase, which breaks down the cellobiose to 2 units of glucose. And this is very important to understand that beta-glucosidase is the rate limiting step, because in most cases, beta-glucosidases are inhibited by its own product, glucose. When there is a glucose concentration above a particular threshold, it starts inhibiting the beta-glucosidase. So the entire cascade is controlled by beta-glucosidase. And the moment beta-glucosidase stops its action, what happens is that there is an accumulation of cellobiose. And in nature, what happens is the organisms that produce these cellulases, they are very tightly regulated by induction and repression mechanisms. The moment there is an accumulation of cellobiose, it starts repressing the other genes, which is responsible for endoglucanase and exoglucanase in the organism. And typically in the world, currently the most highly producing, hypercellulase producing organism currently used for commercial production is a fungus called Trichoderma rhizae. That's C30. So in the case of RATC30, the beta glucosidase of RATC30 is inhibited by 50 millimolar glucose, which is about 0.9%. So at 0.9%, the entire reaction shuts down. We don't want that to happen. We need high concentration of glucose so that we get more of alcohol or more of fermentation products. We need a high concentration of glucose to occur. So what normally we'll do, we take the trichoderma cellulase and add more beta glucosidase into that from maybe another source, a heterogeneous source, so that the enzyme is a blend and it can act with a higher concentration of glucose, attaining very high concentrations of glucose. And another option is you can have glucose tolerant beta glucosidases. Some of the beta glucosidases are not inhibited by glucose or inhibited by very high concentrations of glucose. So if you have an enzyme which is glucose tolerant, a beta glucosidase which is glucose tolerant, you can proceed the reaction to a higher equilibrium of having high concentration of glucose. So this is the mechanism. Again, this is important to understand how the cellobiohydrolases act, because this is just like our nucleases acting on DNA. These enzymes are not just going there, binding, cutting, and leaving. This actually pulls off strands of cellulose, and there is a groove inside this. There are three major portions or domains of this enzyme. One of them is called a carbohydrate binding domain. You have a linker which allows this head part or the catalytic region to move around, and this goes and binds. The cellulose binding domain or CBH allows it to bind on the cellulose, and this linker allows the head to move freely. And this pulls out a strand and it passes it through the groove, and it moves along the strand moves along the groove, and during that process this gets chopped off. The pieces of cellobios get chopped off. So this is how this cellulose hydrolysis proceeds with the cellobiohydrolases. Now, what are the challenges in making these enzymes more viable, commercially, making the whole process commercially viable? Because I mentioned earlier that hydrolysis is the most costly step, and contributing to that is the cost of cellulases. So we have very high cost for the cellulase preparations, and the organisms that produce cellulases like Trichoderma raceyi, they have low cellulase yield. Of course, there are several mutants which have come through several years of genetic manipulations, and some of them are high producers, hyperproducers. Has mostly been done at centers of multinational companies, enzyme manufacturing companies like Novozymes or DuPont, and they have brought up enzymes which are highly efficient, but still the yield of cellulase from these cellulase producers are pretty low, and their efficiencies are, some of them are efficient, but some of them, not all of them, are highly efficient. And then, of course, I mentioned earlier about the induction and repression mechanisms. In most of the organisms, or almost all the organisms which are degrading cellulose, the cellulases are inducible enzymes. They are produced in demand only. Only when the organism is exposed to cellulose in the environment, it starts synthesizing cellulases. And there is a very, very stringent control by repression mechanism, carbon catabolite repression, and there is a controller element called CRE1, which is a major repressor of cellulase genes in Trichoderma. And similar to other, this CRE1 is present in other fungi also. And then feedback inhibition. This also I mentioned. Beta glucosidase is inhibited by its own product glucose, and there can be a strong feedback inhibition of beta glucosidase. Then, of course, the temperature and storage stability of the enzyme. Of course, if you want to perform biomass hydrolysis in very large, huge plants, just like our refineries, we are talking about several millions of liters. And in that capacity, when you are looking at operating in that capacity, you need enzymes in huge quantities. And you need to make these enzymes at some place and transport to the factory. Means the enzyme has to be stable, it has to be thermally stable, and it has to be stable against transportation losses and environment, against the environmental forces, so that the enzymes remain stable and it is, still remains active when it is to be used in the plant. So that means you need the enzyme to be temperature and storage stable. Then, of course, cocktails. I mentioned about trichodermatoses case where it is having very, a beta glucosidase which is low in titer, as well as it is inhibited by its own product. I mean, the Ki of inhibition is 50 millimolar or 0.9 percentage. At 0.9 percentage glucose, it gets inhibited. So the tolerance of the beta glucosidase, that is a problem. So what we, to overcome that, what we typically do is to add enzymes in very high concentrations from some other source into trichoderma. And these other sources may have beta glucosidases, which are slightly more tolerant. Instead of 50 millimolar, it could be 200 millimolar. So it can attain a higher equilibrium of glucose. So how do we start for the cutting of the cost of enzymes? If you are following what is happening in the bioethanol field, the first boost of this thing came in early 70s when there was the first oil crisis, and U.S. National Renewable Research Laboratory started work on converting biomass to ethanol, thinking that this fuel, the crunch, the fuel crunch that happened at that time, the deficiency in petroleum resources, triggered this research. And even at that time, India had very pioneering efforts. And if you look at what has happened at that time, uh, some of the best efforts has happened in India. In IIT Delhi, Professor T.K. Ghosh and his group has been working, and theirs has been the first plant in India for converting, not an integrated plant probably, but it was the first plant for converting lignocellulosic biomass to ethanol. And even today, the enzyme assay methods that we follow, the IUPAC methods for cellulase assay, is from T.K. Ghosh group. This paper is from T.K. Ghosh. But of course, a lot has happened after that. After that, again, the oil prices came down. So people lost interest in this research. And it picked up later in late 2000s, because again, there was an oil crunch. So then NREL had contracted the enzyme majors, Genencor and Novozymes, for bringing, when they realized that enzymes is the major challenge in converting this biomass to ethanol or any other product. So the first thing to do was to bring down the cost of cellulases. So these enzyme majors, Genencor and Novozymes is currently the world's most leading enzyme manufacturer. And Genencor is now taken over by DuPont. And both these companies have brought out enzymes. In the case of Genencor, it is Accelerase. The latest iteration is called Accelerase Trio. And for Novozymes, it is currently called Selexitec 3. So these enzymes are brought, and these are the world's best enzymes currently for biomass hydrolysis. Both of them are cellulase blends, which can work on the biomass, different kinds of biomass. Now, if you look at India, India has now a biofuel policy which I brought out in 2018. And following up that, we are also a signatory of the Paris Convention, and we have been vowed to bring down our carbon emissions to very low levels by 2070. We have targeting zero carbon emission or net zero carbon emission. And one of the major goals proposed during that biofuel policy was that we have to bring down our increase our blend of ethanol in gasoline or petrol by 20% by 2030. So that has been advanced now, and our current goal is to bring down, to achieve that 20% blending of ethanol in gasoline by 2025. So how do we achieve that? We have, of course, our first generation ethanol is there. India is a major producer of ethanol, but mostly it comes from molasses. But that is not sufficient to meet our demand of blending in gasoline to that high levels. So what is needed is a second generation which comes from lignocellulosic material. To do that, we have currently cleared, our government of India has cleared 12 different projects, most of them from our major leading oil manufacturing companies like Indian Oil Corporation, Bharat Petroleum, Hindustan Petroleum, and so on. And 12 projects have been cleared, most of them at 100 kiloliter capacity to produce ethanol from biomass. And most of them works on technology, and some of, at least majority of them works on technologies either imported or from a company called Praj, which is providing ethanol technology, lignocellulosic ethanol technology in India. There are only two technology providers which are Indian. That is, one of them is Praj and the other one is ICT Mumbai. And almost all these technologies use enzymes from Novozymes, Selexitech 2 or 3. So this is where we are, and the cost of these enzymes even now cannot be considered as very economical. Even now the cost of enzyme is pretty high. So we need to bring down the cost of enzymes. So how do we bring down the cost? There are multiple challenges. We can have low cost production technologies like solid state fermentation. I'll come to that and what is solid state fermentation and all. You can use highly efficient cellulases and allied enzymes. We can have better cocktails by mixing different kinds of cellulases and accessory enzymes to create blends, which functions very efficiently, so that you need to use only very less enzymes per unit biomass to be hydrolyzed. And then enzymes with lesser inhibition. Can we have glucose tolerant beta glucosidases? So this will actually allow the equilibrium to go to a higher level of glucose and allow use of lesser enzymes. And another question is, can we reuse the enzyme? Okay, so if we can reuse the enzyme, again, the cost of enzyme will come down. So I'll just highlight some of the efforts that we are doing towards achieving this cost reduction of cellulases. So I'll start with this optimization of production parameters. Means this is just a routine thing. You do a media engineering, you do a process engineering, so that we get higher efficiency of production. So that you can bring down the cost of cellulases. Then you have developing cheaper inducers. Typically, I mean, as I mentioned earlier, this is cellulases are an inducible enzyme. You need an inducer, and typically in nature, the inducers, we still don't know what are the real inducers. But for trichoderma, at least we know that sulfurose is a disaccharide, is considered as a major inducer of cellulases in trichoderma. But sulfurose costs like 35,000 rupees if you look at Sigma catalog for 10 milligram. Obviously, you cannot use this as an inducer for large scale production. So how do we do? I mean, nature, what the organism do for induction is that at the tip of the hyphae, when the fungus is growing, there are beta glucosidase, transglycosylating enzymes, and some basal level activity of cellulases, which breaks down the cellulose. And whatever sugar is generated, it can be transglycosylated to generate disaccharides, which act as inducers. So we can actually use the same mechanism using transglycosylating enzyme to generate a crude mixture of inducers, which is used effectively for producing cellulases. That will bring down the cost of inducers for producing cellulase. Then, of course, on-site production. The advantages of on-site production is that there is no transportation. No cost involved in transportation, no cost involved in storage, no cost involved in concentration. You produce what is required and immediately use in the plant. So this can bring down the cost effectively. Of course, you can have improvement of specific protein engineering to improve the specific activity of the enzyme and to generate more thermostable enzymes. With each higher temperature of activity, the turnover of the enzyme increases really significantly higher. Now you can have genetic improvement of cellulase producers. I will explain some of these things, what we are doing. We can actually have more genes of cellulases put into the organisms to produce that. You can try to address the repression mechanism, block the repression, and maybe shut down some of these transcription factors which are responsible for repression and so on. So these are other methods. Then less expensive feedstock. Instead of using, this is an inducible enzyme, and for producing, typically what we use is pure cellulose has to be supplemented into the medium. So, and pure cellulose sells at the most, the cheapest price of cellulose I found was 200 rupees per kilogram for pure cellulose. So can we go really below that? So if we can use plant material itself, after some processing, if we can use the same plant material as a cellulosic starting material, that can bring down the cost significantly. Then cheaper technology, solid state fermentation. You have submerged and solid state fermentation technologies for producing enzyme, and at least some of you would be knowing about what is solid state and submerged. In submerged, you actually cultivate the organism in a liquid medium. It is mostly aqueous based liquid medium where you add your media components and inoculate the fungus or the bacteria and allow it to grow there. And this requires very highly efficient fermenters with proper controls and so on. And solid state fermentation, on the other hand, is growing this fungus on a semi-solid substrate, solid or semi-solid substrate, along with addition of the minimal amount of moisture which is required for that. So, and this is semi-sterile, and it does not require that kind of infrastructure. So it is easy to grow the fungus, typically fungus, and extract the enzymes from there. So these are just a few examples. We have been using different kinds of raw materials, like, this is wheat bran, this is straw, and this you can see that, different trials done with different kinds of raw materials. And you can see that here in this case, wheat bran gives higher, and then solid state fermentation. Solid state fermentation can give a concentrated product, and requirement of infrastructure is also less. What you see here is the picture of our solid state fermentation pilot plant, and also this is also a pioneering pilot plant with 100 kilogram capacity for producing enzyme. And this plant is going to supply enzymes for our biofuel pilot plant. So this is on-site production, SSF. So this is how it looks like when it grows. These are trays into which the fungus is inoculated on a solid material, in this case wheat bran. After growing, this enzyme can be extracted and directly used. And of course, through process improvements. I will not explain too much here. What I would like to say is that we can actually manipulate the process conditions, the carbon source used, the nitrogen source used, the production conditions like pH, temperature, whatever. You can optimize in multifactorial experiments or single factorial experiments, whatever, and try to arrive at the best optimal combination so that you get the maximum activity. What you see here is the optimization for Trichoderma risi ratsi 30, which is the world's best cellulose producing organism, and Penicillium xanthellum, which is our own strain, CSIR strain. And of course, you can see that this is slightly better than our the world's best producer, Trichoderma risi ratsi 30 also. I will come to that a little bit detail later. So through process optimizations also, you can improve the activity of the enzymes, yield of the enzyme. So these are the organisms that we are working on. For cellulase production, we are using two organisms, Penicillium xanthellum and Trichoderma risi ratsi 30. And for beta-glucosidase production, we are using two organisms, Aspergillus niger and Aspergillus anguis. And these are the average yields that we are obtaining. This is about 50 to 80 units per gram dye substrate for penicillium. And this has been tested in pilot scale, Trichoderma in semi-pilot scale, activity is slightly lesser. And Aspergillus niger again tested in pilot scale with approximately 200 units. And anguis, the advantage with Aspergillus anguis is that it is a glucose tolerant beta-glucosidase. But of course, the yield is very low. I will explain more. I will come to that in a little while from now. And these are pictures showing how the fungus is growing. And initially when it is just before inoculation, and then this is after growth, and recovered after growth and in powdered form. So this is the typical production cycle in SSF. You start with a spore suspension, you inoculate the trays, incubate, and this is after growth. The multibran is obtained, you extract it with water or a buffer, then the enzyme extract is obtained, it's filtered, and you finally obtain the enzyme, and it can be used for hydrolyzing the biomass. I mean, along with cellulases. This is used for blending into the cellulases. Now, why beta-glucosidase addition is important? So what I will show here is an example of why adding beta-glucosidase extraneous beta-glucosidase or heterologous beta-glucosidase is important in making these blends. This you show the typical hydrolysis curve. You start and reach the plateau in about 24 hours. And the sugar concentration that we obtain is 288 milligram per gram. And the moment you add beta-glucosidase, what you see here is you are attaining 321 milligram per gram. And more importantly, instead of 24 hours, you are attaining the peak in 12 hours. So you have double advantage. You're attaining a higher sugar concentration in a lower time. So adding beta-glucosidase can significantly improve the performance of enzymes. And there are, of course, after that, there is a very sharp dip in activity. And that's typically happening because our beta-glucosidase also has a transglycosylation activity. The moment it reaches a peak and the glucose concentration is high, it starts to convert this glucose into disaccharides through the transglycosylation activity. So you have to stop the reaction at 12 hours. But that is fine because you are attaining the peak at 12 hours. So these are some of the studies which we have done with blends. This is a NIIST cellulase. And what you see here is the typical efficiency is 42%. And with 0.2 units filter paper addition, of beta glucosidase addition in the cellulase. Cellulase is assayed typically using a assay system called filter paper activity. You are actually using filter paper. Filter paper is a pure cellulose. So filter paper is used as a substrate. That is why it is represented in filter paper units. So this is the cellulase plus beta glucosidase added. From 42, it has gone to 60%. And with another brand of beta glucosidase added, that is 1.2 units of beta glucosidase filter paper units, it has gone to 60%. Here, what we have added is a Genencose beta glucosidase in a higher concentration. And here is the world's best enzyme. This is Selexitec-2, which is attaining 73% efficiency. So adding beta glucosidase, definitely you can see that is improving the efficiency significantly. But then you have to see that compared to this here and here, the amount of beta glucosidase in the commercial preparation is 300 times more compared to what we have added there. So the lesson is use higher concentration of beta glucosidase to have a starting higher reaction. I will show you the performance figures after some more optimizations. So we have also tried beta glucosidase addition from our Aspergillus niger. Adding to this is a commercial enzyme from Indian enzyme. It's an acid cellulase. And when you add this, the efficiency is going up 33%. But this is not a biomass hydrolyzing enzyme. This is a cellulase used for denim purpose, for denim stone washing. This is not designed for biomass hydrolysis, but the moment you add beta glucosidase, it is performing similar to a biomass hydrolyzing enzyme. And this is an Indian brand, which is a biomass hydrolyzer. There is only one company which manufactures biomass hydrolyzing enzymes in India. And this is that enzyme. And the moment you add Aspergillus niger beta glucosidase, this already has very good concentrations of beta glucosidase. But even into that, when we add our Aspergillus niger beta glucosidase, it is improving 9%. And this is the world's best enzyme. Here also the improvement is evident. There is a 6% improvement, even though this has, as compared to our original enzyme, 300 times more beta glucosidase. Even into that, when you add our beta glucosidase, it's still increasing the efficiency, which shows the importance of this beta glucosidase addition. So what we have done, we have now come across, the importance of beta glucosidase, but this beta glucosidase from Aspergillus niger, for which our tolerance, glucose tolerance is 200 millimolar. But can we go higher? Can we attain higher beta glucosidase? Glucose tolerant beta glucosidase can be obtained from somewhere. So naturally, when we look for biomass hydrolyzing enzyme, where do you look for? Where there is biomass. So naturally, we had gone to several regions in our Western Ghats, and mostly Silent Valley, because that is mostly a virgin forest. And we have covered almost 50 kilometer squares of area in Silent Valley National Park, coming back with several fungi, almost 700 different fungi, and we started screening them. And this has eventually resulted in us obtaining a culture called Aspergillus anguis, which can tolerate 1.5 molar glucose. 1.5 molar is 27%. Compare this with Trichoderma rhizi, which gets inhibited. The Ki is 50 millimolar or 0.9%. And the Ki for this is 800 millimolar, which is 14%. So that much improvement is there. But the caveat here is that it produces the enzyme in very, very low titers. Also, it produces the entire, it has very, very less other cellulase activity. We don't know whether it produces 100, all the other kind of activities, but beta glucosidase is there in low concentrations, secreted in low concentrations, but it is highly glucose tolerant. So what do we need? We need to get this beta glucosidase in very large quantities. So naturally, what would you expect? You have to do some genetic manipulations and put this gene in some other system or the same system with better controls and get higher production of beta glucosidase, glucose tolerant. So before that, we need to understand whether it performs or not. So adding into blends, what we see here, Aspergillus anguis beta glucosidase added into our enzyme, cellulase, gives an efficiency of 83%. And this is world's best enzyme, which is only 78%. So now you see the importance of a glucose tolerant beta glucosidase going into our blend. So only concern right now what we have is how to enhance the production of this beta glucosidase so that we can use this more in our enzyme so that this can actually resolve the issue of the cost of hydrolysis. So we started by looking at different options, and typically, when you try the classical expression systems like bacterial or yeast expression systems, is not very amenable because that is costly. Because even if in Pichia pastoris and all, you need to induce with methanol, you need a very sophisticated system, and for bacteria also this is not very feasible because of the cost involved. So we need to have a natural system which works very well with our natural biomass substrates. So that is why when we started looking at, we did not have too much options for expression system. So we started developing our own expression system. And what we did was to use the same Aspergillus anguis as an expression system. We engineered this Aspergillus expression cassette so that it has efficient promoters taken from Trichoderma, the cellobiohydrolase promoter, which is one of the most efficient promoters in the fungal world. And we put this gene downstream of the cellobiohydrolase promoter. And in this case, this is another example when we started with the GAPD promoter. And these are initial testings where we did with GFP. And what you see here is the GFP expression in this fungus. And we also expressed the human interferon beta just to show that this expression system can handle different kinds of heterologous protein expressions. But even then, our secretion was not so efficient. Even then, our the yield was pretty less. So we really needed to understand why that is so, and whether this organism is really can be used as a host. So that is a study which is ongoing. But of course, I will show you some results from there. We did a whole transcriptome analysis to understand how these genes are regulated in this particular fungus, Aspergillus anguis. What you see here is a self-organizing map study. This is a transcriptome analysis using self-organizing maps. And what we are looking for is genes which are co-expressed along with our beta glucosidase, so that we will understand what are the transcription factors which controls that. What are the factors which control the cellulose expression in this fungus, so that we can manipulate them and maybe enhance its production and secretion. So this is the cluster which contains our gene. Each of these line is a gene. What you are seeing here is 3 different time points: 12-hour, 13-hour, and 14-hour after induction, and whether the genes are expressing. And this is the pattern of our expression of our particular gene of interest. So this is a more enlarged image, and you see our gene of interest and the other genes which are having the same expression pattern. This is our gene, and there are a lot of genes which are expressed along with that. So what are those genes? So what we are really interested in are some of these transcription factors. We have these have seldom been described in any of these fungus before. This is for the first time we are looking at, and that's Aspergillus anguis is a very rare fungus. So we are looking at different transcription factors. There are certain zinc finger domain transcription factors, and then there are kinases. I'm just highlighting just one aspect. This is an exclusive, very, very large data set. Out of that, I'm just picking up a few points to show that there are certain genes which are co-expressed along with our gene of interest, beta glucosidase. And we are primarily interested in the transcription factors and regulatory elements, so that we can actually look at how we can manipulate them to get a higher production of this enzyme. Okay, another story is with the Penicillium xanthinellum. I mentioned earlier that the world's best organism which is used currently for cellulose production is Trichoderma rhizi, strain called RATC 30. And that is used by most of the world's best enzyme manufacturers for producing biomass hydrolyzing enzymes. But is it the only option? For several past 50 years, most of these studies have been happening in Trichoderma, and most of the knowledge currently that we have on cellulose regulation is based on Trichoderma rhizi's system. So, but then we found that our Penicillium xanthinellum, which is actually isolated at one of our sister laboratories in NCL, Pune, and we have been working on its genetics now for quite some years now. But just to show that forms, this is hydrolysis performance in alkali pretreated rice straw, and this is acid pretreated rice straw. In all these cases, what you see here, the performance figures, the yield of sugars from biomass, this is rice straw, is very much high in Penicillium xanthinellum. So why is that so? And the cellobiose is actually lower, and that is good because there is lower cellobiose accumulation. All the cellobiose which is generated is getting converted into glucose, which means that Penicillium gentianellum is much superior to Trichoderma in terms of its hydrolytic profile. But is that all? Why that is so? So we needed to understand why this Penicillium gentianellum is performing better than Trichoderma resi. I mean, we just can't stop there by telling that this is performing better just based on few studies where we show the hydrolytic profiles. So we sequence the entire organism, and this is deposited here. Anybody can go and access this information also. And what we find here, we then aside the major cellulase activities. We see that, okay, here the total cellulase activity, endoglucanase activity, and beta glucosidase activities are the critical activities of cellulases. Is much higher for Penicillium gentianellum, especially for beta glucosidase. If you look here, there is a huge lot of difference between Trichoderma and Penicillium. And then these are the different classes of proteins which are expressed, which are found in Penicillium gentianellum against Trichoderma. And what you see here is cazymes. Cazymes is the carbohydrate active enzymes, which are responsible for cellulose or the digestion of any carbohydrates. You have 27 different, genes located for carbohydrate active enzymes against 20 in Trichoderma. And of course, this difference is there in almost all the other categories as well. Now, even further, if you look at what are the different kinds of cazymes, then you see that these are glycosyl hydrolases. Glycosyl hydrolases are primarily the class under which cellulases comes. Cellulases or any other, most of the sugar polymer digesting enzymes comes under glycosyl hydrolases. So if you look here, you have 26 glycosyl hydrolases for Penicillium against 14 of Trichoderma. And similarly, if you look here, even within that glycosyl hydrolase families, family 7 is basically endoglucanase and exoglucanase. You have family 6, again, is exclusively endoglucanase and exoglucanase. Okay. This also can have chitinases and other things. And you have glycosyl hydrolase 5 and glycosyl hydrolase 3, which under which class the beta glucosidase comes. So if you look at all these different categories, you can see that the amount of beta glucosidase, if you see in Penicillium gentianellum, is very high. You have almost 22 different beta glucosidase genes represented in Penicillium gentianellum against maybe around 3 to 5 in Trichoderma. And endoglucanases, there are 9 endoglucanases listed and 3 cellobiohydrolases listed for Penicillium gentianellum. And these are the comparison, different classes. If you look here, these auxiliary activities, cellulose binding modules, carboxylesterases, glycosyl hydrolases, glycosyl transferases, polysaccharide lyases, all these, everything is higher for Penicillium gentianellum. And then, of course, if you look at the activities of cellobiohydrolase, endoglucanase, and beta glucosidase, again, this is higher for penicillin. And we also looked at the transcriptome analysis of induced and induced glucose grown and cellulose grown cultures. And what you see here, again, these are the difference in expression, very highly induced. All these genes are very highly induced. We can see that beta glucosidase here, this is almost 70-fold. This is log-fold. So this is 70 times higher expression. And these are again 80 to 70 times higher expression for cellobiohydrolases. And these are almost 60 times higher, 60 to 50, 40 to 60 times higher expression of all these enzymes. And then you also have several non-regulators, ARA-A, then CRE-A is the equivalent of CRE1 in Trichoderma, which is a negative regulator, which is down-regulated. XLNR is a central regulator in Trichoderma. That is also upregulated. Then you have ACE1, which is a negative regulator. So if you look at all these things, these are all present there, and it is similar pattern of upregulation of downregulation is happening in Penicillium gentianellum. So, but then is that enough? Whether these regulators have their, do they really act on our enzymes? So we did a promoter analysis of cellobiohydrolase, endoglucanase, and beta glucosidase, and you can see that for all these regulatory elements, there are binding sites on the promoters of these major enzyme classes, which means that these are under the regulation of the transcription factors what we have obtained. And very interestingly, These are some very novel transcription factors, which has not been described before. And the most interesting thing about this is this particular transcription factors, which is expressed almost 100-fold higher. And this has a major major facilitator superfamily signature along with this transcription factor signal. It can at the same time be a sugar transporter as well as a transcription factors, which has a lot of implications in how the signal is transduced from the cell surface to the nucleus. So we are currently studying this, and once we have this information, probably we are thinking that these manipulating these can really improve the cellulose production in this fungus. Okay. Another thing I mentioned about solid state fermentation and submerged fermentation. Is there any advantage of doing solid state fermentation? And interestingly, I believe there is. We did, these are the performance figures. In this case, the sugar release is higher, slightly higher for solid state fermentation. And basically the pentose sugar, xylose release is much higher for solid state fermentation compared to submerged. And if you look at there is a 2D gel electrophoresis comparing both these solid state and submerged fermentation. And secretome analysis shows that apart from the classical cellulases, which are present in solid state and submerged fermentation commonly, there are several genes which are highly expressed only in solid state fermentation, which means that it is not just a concentration effect. The quality and quantity of the proteins which are present in solid state fermentation is much higher. So other things that we are doing is basically to do the optimizations of the cocktails using neural network studies and so many other different artificial intelligence methods, where we collaborate with our materials division and computational modeling division to arrive at cocktails and how to predict how these combinations of enzymes can be derived so that you get the most efficient enzymes. So these are figures showing how the hydrolysis proceeds. This is the starting material, pretreated biomass. This is just after adding enzyme, 1 hour into the hydrolysis, 24 hours into the hydrolysis, when it is becoming very kind of a Newtonian fluid. So I will stop in a couple of slides more. So what are the strategies that we look into improving the enzyme cocktails and how to improve the efficiency of hydrolysis? There are enzyme-related factors like type and composition of the enzyme, what enzymes that you are using, what is the combination of enzymes that you are using, what is the concentration of enzymes in the reaction mixture, how much enzyme you have to load. If you can reduce the concentration that you're using, of course, you can have a price advantage. Then non-productive binding to lignin. This aspect I did not cover because it's too exhaustive. Because lignin, actually, when you're pre-treating, the lignin can get dislodged from the biomass and it can reabsorb onto the biomass. And when there is lignin on the surface, it again prevents the enzyme from absorbing onto the biomass. So there is a non-productive absorption. So you can use surfactants into the enzyme mixture to prevent this non-productive binding. Then inhibition by cellobiose and glucose. You have to tackle this inhibition problem. Multiple methods, as I mentioned before, using glucose and beta glucosidase or something. Then there is a loss of activity by shear and temperature when you're using in reactors. So you have to modulate the reactor conditions, the impellers and other things, so that you get the best reactor configuration for attaining this maximum efficiency. And of course, there are other inhibitors which comes from lignin and hydrolysis of pentose sugars like aromatics and perfurals. You can do a detoxification. And then you also have to have the optimal conditions of activity like pH, temperature, and other things. And of course, I mentioned about the surface active agents, which are to be added into the enzyme mixture so that there is no non-productive binding. This is just to show a figure how the cost is calculated and how one of the means by which we can, I mean, some of the figures are hidden because some of these are proprietary information. So what you can see here, cellulose contributes maximum to the final cost of the media components. Out of the entire 60 rupees of media components, 50 rupees comes from cellulose. And on the processing side, if you look here, the solvent contributes 40%, and you have air conditioning charges and other things. And altogether, per kilogram of enzyme, you are estimated a cost of 245 rupees. Even 245 rupees is not sufficient. You have to still bring it down to maybe 50 rupees or 60 rupees. And one way to do this is replace the solvent by a membrane separation. We already have now started that, and the data shows that it is very highly efficient. And there is significant reduction from 40, we can bring it down to below 10 rupees or something. We already have installed a ceramic membrane unit in our system to do this concentration instead of using solvent precipitation. This is just a comparison of basically commercial enzymes available in the Indian market, because it is not just enough that we work with our enzyme. So there are a lot of Indian companies which manufacture cellulases. But why are they not able to get into this business of biomass hydrolyzing enzymes? Because they are lacking something. So most of these has cellulases, but they may not have the accessory activities. So blending in of some of these activities might help to develop enzymes in the Indian market itself. So we have evaluated several major Indian manufacturers and compared it against the best, Novozymes. And this is another Indian manufacturer who manufactures biomass hydrolyzing enzymes. You can see that some of these companies, the enzymes from some of these companies are performing as good as probably our Novozymes, at least to some extent. So if you find out what is lacking there, we can actually try to blend in maybe enzymes from different companies and bring them together. And it is in nation's interest to have cocktails which are produced indigenously. So these are some of the activities currently ongoing, and we are trying to actually profile the proteome from these preparations to see that what they contain and what they're lacking, so that we can mix and match. And there is a decision-making system also in pipeline where we will be employing some neural network-based or artificial intelligence-based method, so that if you feed an enzyme's proteome profile into that, you can actually predict how they perform and how with this particular company's enzyme, what other enzyme has to be blended into this so that it can be made more efficient to hydrolyze a particular kind of biomass. So I will stop here. These are different things. Again, it will be a repetition. There are different strategies which you can use. You can engineer enzyme to have better binding sites. These are genetic modifications possible. Then thermostability, again through genetic modification. Then disallowing non-specific binding. Again, it can be performed by adding cellulose binding modules to the enzyme's structure. And of course, you can still prospect for naturally better enzymes. We are still we have still explored very, very less of our biodiversity. If you really go into this, probably there are still much more naturally better producing organisms waiting there to be discovered. And then tailor-made cocktails, and of course, understanding our reaction kinetics will go a long way to understand how these enzymes can be improved. And external agents can be added, of course, surfactants and other things. So I'll stop here. Thank you all for giving me an opportunity to speak here. Yeah, this is our pilot plant. The entire process, starting from milling, pretreatment, solid liquid separation, hydrolysis, distillation, alcohol fermentation, and distillation. And this is, I thank all my group members for this. Thank you.
Thank you, Dr. Rajeev, for the excellent lecture. Before I conclude, let me ask the audience to ask questions. Dr. Rajeev has talked to us for almost an hour. I've given you a lot of information, and I'm sure that there should be some inquisitive questions coming from the audience. You should ask. You must ask.
Noxious weeds or invasive weeds are a threat to the environment. How far is the possibility of using these as the substrate for the cellulose production?
I would say yes, as long as you can have, as long as the wheat has enough cellulose and hemicellulose, probably it can be used. But there has been some hype about using aquatic weeds. Only in those cases I have a little bit of apprehension, because it is almost 90% water. So the cost you spend on recovering that biomass and what you get from there will not match. Because when you are spending for taking out 1 ton of material, you're actually getting maybe 100 kilograms of it. dry material. So apart from that, any terrestrial biomasses, most probably yes. Only there are of course concerns of logistics, because how to harvest them and how to get it in enough quantities. You're talking about 100 kiloliter per day capacity and all. Per plant will be utilizing almost 1,000 tons per day. So 1,000 tons of biomass getting itself is a challenge. So that is more suitable for agro residues.
Then about the
Yeah.
getting enzymes, how
Yeah.
far this genetic modification work is going on in our country? Or outside?
There are several groups working on different aspects of cellulases and other things. And of course, our group has been working on, at least as far as I know, from the moment I joined NIST, our group has been working on this. Of course, there are other groups also within CSIR laboratories, outside CSIR laboratories, universities, and so on. But commercially viable technologies, I believe, still are yet to come.
Thank you, sir.
The reusability of the enzyme.
I really missed the telling that. Now, why it is not reusable? If you remember, I mentioned about the modality of its working. You have, if you remember one of my slides in the beginning, where I showed how the enzyme works. The exoglucanases actually pulls out a fiber, and it moves along the fiber. That is how it chops off pieces. Now imagine doing that, exoglucanase on an immobilized thing. It cannot really make a free movement. So it is not really amenable for immobilization. So that is one of the reasons why immobilization cannot be a technology for reusing the enzymes, or except for beta glucosidase. But that is one of the most common methods of reusing. But other than that, maybe you can try thinking of reusing the enzymes through membrane separation and putting the enzyme back in there, and then maybe recycling the undigested residues with adsorbed enzyme back into the system and so on. But all these systems have their own limitations. Some of them will add the cost. Some of them are practically very challenging, especially like putting back the residues, because that residues will again add on to the bulk of biomass, creating actually mixing problems and mass transfer problems.
Expression level is greater. What may be the reasons?
The substrates used in SSR are complex. You have lignocellulosic material like straw, for example, or you have wheat bran or similar that kind of material, which are not just one thing. When you're using solid submerged fermentation, at least in our case, we are using pure cellulose. In this case, we are using a mixture of so many different things. So each of these things might be inducing different kinds of systems. So the organism might be responding in a different way. So the quality and quantity of proteins which are expressed are different. That is what we observe now. We have studied only with the rice straw and with the Penicillium gentianella, but we are expanding that study now.
The student side. It's very difficult to instigate them to ask questions. Anyway, Rajiv, I have a few queries to make. One is that bioethanol production using all these kind of biomasses, which are rather unused or waste materials. Now, compared to the normal ethanol production using molasses, what would be the product? I'm sure that would be cheaper compared to that. But then you are using some material which is going waste into a useful product. That for that matter, even if it is little high, it's okay. I agree. But what I say is that molasses, when they are used, there is a lot of waste material that's coming out. Could you use your technology to recover those wastes coming out from the ethanol producing factories, so that you will have coupled your technology to that of the normal ethanol production?
Hybrid thing with first generation and second generation together. Sir, I would say that first generation can take care of itself. We really do not need to couple this there. because their residue is basically after molasses makes basically a sugary residue with basically what you call as a vinasse. After fermentation, all the yeast cells and other things will settle down along with the molasses residues and other things, and leftover sugars plus yeast cells and other things. So that is more suitable for a non-enzymatic anaerobic conversion, just like your anaerobic digestion to produce biogas and other things. So that is the most probable and most suitable solution for that. I mean, most of the current distilleries are using that. Now, at least in our country, we have a regulation that we cannot discharge. These distilleries should be zero discharge. So they have to take care of this, and they are currently doing, at least on paper, they are doing.
Yeah. Another question is that compared to the biodiesel production is very, cheaper way of making diesel.
Yes.
Because wherein only a esterification process is involved. That's why people are focusing more on the biodiesel production, because the cost effectiveness can be really brought in over there, and it's also a biofuel, which can be used for diesel engines. How do you survive the competition between the biodiesel production using the bioresources and your bioethanol production?
Sir, actually,
Do you get much of
yes.
support?
No, I mean, in India at least, biodiesel again, similar to ethanol, is facing a lot of challenges because we do not have raw material. Just like our, petroleum, we are also a net importer of oil, edible oil. We do not have, enough oil in our country for our edible purposes. We are actually importing oil. So biodiesel, when it is finally formed, after the entire process, you cannot really sell it for a cost like, which is competitive with the petroleum diesel. So at least 70 to 80 rupees per liter would be there, because even if you start with a non-edible oil like Jafrofa or something, you have to spend per kilogram of Jafrofa, you are spending 25 rupees. And from that, oil content might be 35%. So in the end, even the raw material itself will be 50 rupees. And after transesterification and other processing, you add together all the logistics, everything. It will be really hard to, make it really economical. Maybe
Microalgae.
Microalgae is even more challenging because the yield of microalgae from per liter could be at the most maybe 5 gram per liter, and out of that only 20% is oil, which means per liter you are getting only 1 gram of oil. And to get several thousands of liters of oil, you need kilometer squares of area for cultivating these algae if it is on an open pond system. And even in when you are doing something like a fermentative kind of production with anaerobic or whatever, so by cultivating algae in terms of that's a non-autotrophic method, heterotrophic method, you still have to supply glucose or any other raw material harvesting.
No, I mean only photo bioreactors.
Then it is, as of now, it is kind of very, very difficult, very challenging because of the kind of cost involved in producing the algae and separating it. Because to get 1 gram of algae, you have to process 1 liter of water.
So finally, what are the challenges and opportunities with your technology?
I would not say that any of these technologies is mature as of now. So it should be a combination of different things. I mean, there are very, very interesting things which are coming up. Say, for example, I see that 12 different projects have been cleared in our country. And one of the promising, according to my belief, Numaligarh Biorefinery is coming up in Assam, which is using bamboo as a raw material. And this is based on a technology from a company called Chempolis in Finland. They are pioneers in wood processing and other things. They have a pipeline for separating the lignin, value adding the lignin, and C5 to purpurals and other things. So it's like a biorefinery. So only those aspects, I mean, those situations where we are operating in a biorefinery, then only this is going to be economical. So directly biomass to ethanol alone is not going to sustain. So the cost is too high. And similar thing with other kinds of systems also. Algae, we have to think about can we use some other value addition so that this cost can be offset with something else.